Welcome everyone to our webinar, Applying AI to All Stages of Drug Discovery and Development, the Impact on Patients' Lives. I'm joined today by Sylvie Laquerre, VP of Oncology Therapeutic Area at Janssen R&D, and Caroline Ferris, our Senior Director of Health and Life Sciences at Domino Data Lab. I hope everyone enjoys what we have prepared for you today. And with that, I'll hand it off to Caroline to kick off the discussion.
Hello and welcome, everyone. My name is Caroline Ferris, and I work for Domino Data Lab. Domino is an enterprise data science platform which allows companies to scale their data science capabilities and unleash the power of data science in order to solve the world's most challenging problems. Here at Domino, I lead the health and life sciences strategy. And Janssen Research and Development is using Domino to actually power and scale the use of data science across their R&D. And I am really happy to have with me here Sylvie in order to see what impact that is actually having. So welcome, Sylvie. Could you please tell us a little bit about yourself and about Janssen?
Thank you so much for inviting me. I am not an expert in artificial intelligence, but I'm certainly using it very often in drug discovery at Janssen. So my name is Sylvie Laquerre, and I'm leading the solid tumor targeted therapy at Janssen. And Janssen is the division of R&D of Johnson Johnson. My role, in this role, I'm responsible for the strategy, and I'm also responsible to bring target from target identification to proof of concept in the clinic. So everything is in solid tumor is coming into my group. And I'm very pleased today here to talk to you on the artificial intelligence. And I think you wanted to tell you a little bit about my history. Is that I'm at Janssen now for about like 9 years. I was at GSK before for 10 years, and I was working in oncology there also mainly. And before that, I was working on both coasts in a biotech company. And if you wonder where the accent is from, so you can stop guessing where I'm from. I'm from Quebec City, so that's where the French accent is. I came here for a postdoc at University of Pittsburgh, UPMC. I thought I'm going to stay a couple of years, and 30 years later, I'm still here. So when I gave up, the accent is not going away. So that's what you have.
Thanks, Sylvie. So from my side of the world, we've experienced an increased interest in pharma for AI/ML. Why would pharma be interested in AI/ML?
Well, in my opinion, and obviously everything is in my opinion. So I think that there's two main reasons why I think pharma is interested, that I am interested in AI machine learning. One of them is the quantity of data and the diversity of data that we have to handle and we have available to us. And it's coming from all sort of source of information. And we know that when it's all different information, that it's very difficult for a human brain to computerize. So that's why we need artificial intelligence and machine learning. To give you an example, the human genome is about 3 billion nucleotides. So in late 90, early 2000, it took about 13 years and $2 million to sequence the entire human genome. Well, today we can do that in about a week for $1,000. And we do it. And we have a lot of information coming from thousands of patients, thousands of genetic information, and also with their prognostic, their response to treatment. And all of that need to be understood in a very comprehensive way. And there's no way a human brain can do that. So we use artificial intelligence to better understand that. The other reason why I think it's important to use machine learning and artificial intelligence is really in a question of saving money and time, and to also be competitive in a way to bring the best drug in a faster manner to our patient. In a way to save time and money, it's just because we are a very regulated industry. So the FDA is really managing what is going to be approved. Like if you have toothpaste, you can say that you have the best toothpaste on the planet, and you don't have to prove it. You just put it on the shelf and you sell it. But if it's a drug that is a prescription drug, you have to show that it's better than the previous ones that were there. So it's always more and more difficult to discover drugs because there's always some more that are coming on the market. So we are competing with other people to bring drug to the market, which is beneficial for the patient. And in discovery, we don't make big leap. It's always a small incremental improvement that we do over each other. So we have all about the same data. So it's how do we manipulate that data to get that information that is going to give you this little leap or this little improvement to make a better drug, to bring it faster and more efficiently to patients. Just to give you an example, in drug discovery, it takes about 12 years to bring a drug to the market. So we need to accelerate that. It's not sustainable this way. So we need to accelerate the process, and artificial intelligence and all the data that is available is really helping us in advancing better drug faster for the patient.
That's great. So AI, basically how well you do AI is your competitive edge in pharma, essentially.
That's correct. That's correct.
So at which steps of R&D then are you using AI ML most? And can you give us some examples of that?
Well, I think that we're using it at pretty much every steps of drug discovery. And I'm not saying that a drug will make its way through AI at every step of the process, but I think that we have the potential to use it, and depending on what we're looking for, we're going to use AI at different places. So if I give you examples, I can start very early because that's where my heart is. I'm a drug discoverer, so I like a lot of discovery. I think one of the most important things in drug discovery, it's probably to select the right target. So which target are we going to take in the disease that we want to inhibit this target? So that means that the target has to be a driver, something that will drive the normal cell to become cancer. And for example, I can talk about like lung cancer that I know very well. So in lung cancer, when you have a mutation in the epidermal growth factor receptor, or EGFR, the cancer cell, when it's mutated in EGFR, it become cancer. So if you inhibit EGFR when it's mutant, that will kill the cancer cell, and that way that will make an advantage to the patient. So clearly, EGFR is a good target. But you can imagine a lot of people are working on EGFR. There's a drug that are approved against EGFR. In fact, it's the standard of care. If you have lung cancer and you have EGFR mutation, you're going to get a drug against EGFR. So, but that represents about 25 to 50% of all mutation in lung cancer. So how do you go after the other mutation? How do you identify the mutation that you can go after? Well, we have genomic information that is provided from sequencing tumor, and then we can find which one is mutated there. And that's about 50% of lung cancer. So there's 50% that we don't really know what is driving the cancer. So that's where we apply some artificial intelligence there. So what we do is that we take tumor from patient and we sequence them, the entire genome, and we also do the RNA, the entire transcriptomic. So that's a lot of data that we are looking at. And we reason, and a lot of the people are doing that, is that what two genes are never mutated together. So that means that if they were to be mutated together, that will cause cancer death. So cancer have evolved not to mutate those two genes together. So you can imagine that there's a lot of potential combination. There's over 30,000 genes in each cell. So if you're looking at all the permutation, it's impossible to figure out which one are never mutated together. But through machine learning, we can really identify which pairs are never mutated together. So that way, instead of having, 40 billion combination, we probably have 100 of them that now we can test in the lab and define which one we can go after. So that's what we've been doing. And now we identify a unique pair, and we're going after one of the target to address lung cancer. So it's a little complicated maybe, but the principle is that artificial intelligence is really helping us to identify target that the human brain would not be able to disseminate from the entire genomic sequence that we have available to us.
So I have a quick question about that, because it sounds like you mentioned in your example a very typical cancer, like you said, 50% of the lung cancers are of this type of mutation. Is machine learning allowing us to be able to invest, because it costs money to invest in these things, invest in cancers that are not so typical, so that patients ultimately that have more atypical types of cancers have a greater chance of getting medicines to help them?
Absolutely. And, we often use cancer that we have a lot of material, like lung cancer, we have a lot of biopsies and we have a lot of information. But when we find something, cancer doesn't reinvent itself for every type of indication. We can see if it applies in smaller population, in more rare disease, in more rare type of cancer. And if those signatures are applying there, then we can certainly develop a drug for those indications.
That's
and
great.
In fact, every time that we test a different indication, every time that we test a new set of patients, we enrich our database and we become like a test set versus a training set. And we can really exploit a little bit more the capability to address different cancer. Sometimes we find that mutation in lung cancer is very similar to mutation in head and neck, for example. So we can apply those mutations that we find, or those targets that we find in lung cancer to other type of cancer. That's a good question.
Oh, that's great. So you've identified the target, and what is the next step then in innovation to finding a new medicine?
Well, when you have a target, you always have to validate it to make sure that it is a good target. But then after, you have to find a way to inhibit this target. So you need a small molecule or a drug that will prevent the function of this protein. So we call that screening to find hits. So most pharmaceutical companies have collection of compounds that are representative of different drugs, about 1 million of them that we have in our collection, plus or minus 1 million. So we can test those drugs one by one to see if they are inhibiting our targets. But if you're looking at the potential of how many drugs you can make, if you're looking at the carbon, the nitrogen, and the oxygen, what are the potential of making drug? It's 10 to the 60. There is no way we can screen 10 to the 60 potential molecule in the lab. So what we do, we use artificial intelligence to really look at the probability of a drug to bind to a target. And we call that virtual screening. So we have virtual compound collection. It's all in the computer. And we bring our target. We have a structure of the target. And the virtual screening is taking one molecule at the time and is trying to see if it's binding in the pocket of the drug of interest. So at the end of the day, we might have 200 molecules that have a good fit. So if you have 200 molecules, it's much more manageable. But we tested billion virtually. So we have about 200 molecules that we can now go and make. The chemist can go in the lab, make those molecules, and then the biologists and the biochemists can test them. And then that's what we call that a hit. We have a compound that blocks our protein of interest.
So you have a hit now. How far are you from actually testing in the clinic?
Well, quite far. So it takes about, I would say at that point, maybe between 5 and 7 years to get to the clinic. And it's just that a hit is a really primary, very early step in drug discovery. So the hit is probably not bioavailable, meaning that if you take it as a pill, it's probably not going in your system. The pharmacokinetic is probably not right yet. And it's probably not being metabolized properly. And the biggest problem that it probably has, and I'm pretty sure it does have, it's probably toxic. So it's really important for us to find compounds that are active, but are also that are not toxic. And toxicology is another place where we apply artificial intelligence. And the way we do that in drug discovery, we work a lot to identify hit, and then we make them better. And one of the last steps is to look at the toxicology, make sure that the compound is active at non-toxic dose. And most of the time, it's very late in the process. It's probably like year 6, 7, or 8 that you're doing toxicology. And we do it this way because it's very time consuming. We do one compound at a time, and we put that in animal model, often rat, and we're looking at the toxicity. So, and I would say that probably more than 50%, if it's not 70, 80% of the compound fail at that stage because they're active, but they're also toxic. So then we have to go back to square one. The compound is toxic. We have to go back 5, 6 years earlier on, and we go back and we bring another one. So it's really time consuming to do that. So by putting artificial intelligence very early on to really understand if a compound is toxic using database that we have is really going to help us. And one way we do that by high content imaging. So we know what compound, if they're toxic or not, because we tested them before. And then we treat cells with those compounds, and we put marker on cells, maybe 40 or 60 different markers that are colored differently. And based on what this compound is doing on those cells, the cells are going to have a different pattern of luminescence, if you want. We call that cell painting. So they're going to be painted differently. So we're going to see what is the toxicity. Liver toxicity has a different high content imaging than neuronal toxicity. So now that we have those tools that are being developed through high content imaging to image learning, we can really go early in the process and test 2,000 compounds very early on and take a structure that has the less potential of being toxic and advance that earlier on. So this is very transformational of how we can really detect toxicity of compound way earlier on in massive quantity of compound versus one compound at the time late in the process.
So you said it was like 5 to 7 years. This process was 5 to 7 years. Is what you described using machine learning to test earlier? Is that going to reduce that time? Is the 5 to 7 years, is that where we are now? Or if you apply machine learning, how much time can you actually reduce?
It's really one of the goals, as I told you, it's to reduce and being more efficient. And I think that if you take a compound that is toxic on year 5 and you have to go back 2 years, well, you'll waste 2 years. So maybe with machine learning, we can save those 2 years. And the one that we're bringing in later stage of testing are going to be pre-tested through machine learning. And we're going to save those years before we bring it into the clinic.
Yeah, and years actually means a lot to the patient.
Absolutely. Often we even think that the patient, even some tests that we have in the clinic that might take 2 to 3 weeks to get the answer, it's too long. Sometimes patients in oncology, they don't have 2 to 3 weeks. So we really have to be fast at delivering drug to the patient. And if you're thinking about lung cancer where you have 2 million diagnoses every year. So that means every day there's people dying of lung cancer. So longer I take time to put a drug in the clinic, less life I can save. Because we're always against the clock, but it's mainly against trying to save patients, to bring them. And maybe sometimes what we give the patient is not necessarily a cure, but might be a way that they can live longer till another treatment that will give them the potential to go into another clinical trial. In fact, we have one of our patients in heme malignancy that went to 3 different clinical trials. And each treatment prolonged his life to be able to have access to a different clinical trial. And now this patient has been having a CAR T, which is cells that are engineered, immune cells that are engineered to kill cancer, and is in full remission. So that's quite inspirational when you have those stories about acting fast to deliver drug to patients to really prolong their life.
I think that's actually very important to know how, and to see how clinical trials also can buy some time before a cure is actually available. So, yeah.
As I was telling you, we, and machine learning is so important there. I'm having an example right in my head that is coming when I'm telling you about that. We, when a patient is diagnosed with cancer, most of the time it's through an X-ray, a CT scan, or an MRI. But the person who's really going to diagnose the cancer is the pathologist. So that means that the patient will have to have a biopsy, and the pathologist is going to look at it and define what cancer the patient has. So that's only to define what is the cancer. But with machine learning, you can take this simple H&E, which is a type of staining that you stain the original cancer. And with machine learning and with all the data that we have on the effect of drug, we can, only with this slide, be able to define what is the best treatment for the patient. And we do that currently. We really are training artificial intelligence of high image content to, just from the pathologist's slide, we can define what is the best treatment for the patient. And every time that we have that and we have the data from the patient, we enrich our database, and it's self-propelling that we are getting better and better at defining ways to treat patients. And we're also accelerating the process. For example, like in breast cancer, one of the genes that is upregulated is called HER2. And if a woman comes with breast cancer, then we need to find if it's HER2 positive or not. If it's HER2 positive, there's treatment available for that. But in order to do that, we need to take that original biopsy, make a slice, put it on the cover slip, send that to a lab, and the lab will do the analysis to see if HER2 is present, send the data back to the clinician, and then the clinician can decide which treatment you have. But now with the high content imaging that I told you initially, the image is available on the computer. We can train the computer to define if HER2 is present or not, no wet lab required, and in about 5 minutes, the clinician can know which treatment to give to the patient. So we cut 3 weeks just like that.
So, you know what? It must be incredibly satisfying to know that the job that you do every day is having that kind of an impact. I would imagine. So what do you think about, like, when you go to work? Is that something that you consider? Is that motivate you?
Well, I think that, I think it's not only me, it's a bit all of us, right? We're all contributing in changing patients' life. It can be from the treating physician to the lab person who discovered the drug in the lab, or the engineer that built the algorithm and analyzed the data to find the best target, find the best hit, and find the best way to provide a diagnosis or a treatment to the patient. So we're all contributing to that. To be honest, every day that I go to work, I probably think more about what is my next meeting in 10 minutes and the document I have to review by the end of the month. But in difficult time, when nothing works and it's a little bit more difficult, then I really get a lot of my inspiration from testimony from the patient. And being in the industry, we are privileged to have access to that. And there's always stories that are very close to my heart, that I've seen patients that have benefited from our treatment. And I will always remember when I was at GSK and we discovered this drug for melanoma. It's a combination therapy of a MEK and a BRAF inhibitor. And this father, all his life, was looking forward to walk his daughter in the aisle for her wedding, but he had exhausted all potential therapy for his cancer. So he came onto our clinical trial, and we gave them to the treatment the opportunity to go to his daughter's wedding. The joy that we provided this man to be able to do that is priceless. And I get a lot of my inspiration in this kind of testimony. And I just wish that all of you can have access to that, because it's very inspirational. And I hope that when you're in front of the computer and you find also that it's depressing, well, think of that story, because we're all a little bit part of that.
I know how you feel, Sylvie, and I'm not a scientist. I'm not even close to the end patient, but I would like to think that what I am doing in some way is helping people. And I feel like that's something that drives me as well. So I have back to AI, ML. Do you?
Oh yeah, that's why we're here, right?
Yeah, that's why we're here. So tell me about where you see the future of AI/ML serving patients at its best.
Well, I think that in the very close future, it's almost now, we have access to all that genomic information. And we have every biopsy is going to be fully sequenced, very close. We're doing that now. And every biopsy is going to be image content. So I think that in the very short future, if it's not now, all that information is really helping what the patient treatment can have, what kind of the best treatment for the patient. So I think that that's a little bit where I see that it's going on, but there's also a lot of data that are becoming available. You know, we have all those devices that are attached to our wrist that we are collecting. And then now we're not only seeing like what it is that the patient is taking as a drug, but it also what is influenced the treatment paradigm. For example, like a patient might have to walk 10 steps to go to his apartment when he's on treatment. Other patient might have a high protein diet. Other patient might exercise and some don't. So with the information that we have now that is collected, we can probably all put that together at the same time that we do the clinical trial to really like assess not only what the drug is doing, but the behavioral of the patient during the treatment that is accompanying the patient. I have a supervisor at one point who told me that he was doing a mouse study, and the graduate student just opened all the cages of the mouse before he left for the weekend. And the mouse went in the basement, and then some of them went in the cafeteria, and some of them got trapped. So on Monday, the graduate student gathered all the mouse, put them in a cage. That's a clinical trial. So everybody is going home and doing something different, and we don't know about that. So maybe eventually when we have all way to document and collect that information, it's really going to be really informative about how best to treat patients and give them the best treatment.
Thank you so much, Sylvie. I think we have a few questions that people have put in the chat. Maybe we should go through some of them. Can you foresee any additional types of data that will help us to diagnose and treat patients more successfully? And I think the example that you just gave probably answers that.
Yeah, and I think there's also other data. You know, I'm talking to you about the genome and the tumor sequencing, but you know, we're getting more and more information about the microbiome also. And this is a whole set of data that are getting available to us. We know that the microbiome, it's all different for all of us, and it changes in your lifetime, depending if you're taking antibiotic, what you're eating. And we know there's more and more data to show that the microbiome is influencing not only on your disease, but on your response to treatment. So I think that you know, that information of the genomic combined with the microbiome, combined with the wearable that are going to give you information on the patient's behavior is really going to help us predict the future of what's going to happen. Even sometime when I look like, you know, maybe in, you know, 20, 50 years from now, I can see that with all that information, we will be able to maybe visualize our health in the future. I know it sounds crazy, but in a way, we are going to be able to have information on what we can change, behavioral changing, or which drug can we take to prevent us to have cancer. If you're looking at cardiovascular disease, people take drug when they have high blood pressure to avoid heart attack. We have to be the same thing in oncology. We have to find markers that are going to tell us that you're at risk for certain cancer. And I think in the future, we'll be able to do that. And we'll be able to see ourselves, if we continue to have certain habit or do certain thing, what kind of cancer we're going to have. And then we can correct the path before we get them, the same way that we prevent having heart attack by taking different drugs. So in a way, you know, maybe we'll be able to live for another, like maybe living 300 years is going to be possible. Better have a good 401k, but maybe, you know, that's going to be the possible. And that's going to be maybe through data analysis. We don't know how to do that, but with all the information that is present, I think we might be able to get, that's my goal at least.
One question, what are some of the organizational challenges that you have successfully overcome using AI/ML in drug discovery to accelerate the traditional processes? And additionally, what challenges have you likewise overcome with the FDA?
Artificial intelligence is as good as the database you have. And I think one of the challenges that we have is always to get better data. And I think that the FDA is there to help us. The FDA wants us to bring drug to the patient. And they're much more open now about artificial intelligence supporting some of our clinical trials. For example, normally in clinical development, you have to use a control arm, which we call placebo, and a treatment arm. But if you have enough information on the patient data that you don't have to do a placebo arm, which makes sense to me, that why not giving the treatment for patient, the FDA is open to that. If the data are there to support that you only need an active arm and not a control arm, they will accept your data to move forward. So there is challenges, but I think everybody it's there to stay. Everybody is aware that artificial intelligence is really helping us to advance the science in a very precise manner. So we do have challenge, but I think that everybody is trying to work together to solve them. But more data, more information is always something that we need.
Thanks, Sylvie. So we actually have a couple of questions here that are really patient focused. I actually have these questions myself. So what would you recommend someone do to find a clinical trial that might work with their cancer? I know this is important for a lot of people.
When people are diagnosed with cancer, usually they will, it's a pretty well-defined treatment regimen. So they will go to the standard of care. Like I told you, if you have lung cancer and you have EGFR mutation, you're gonna get an EGFR inhibitor, and that's well known. It's when you progress into the current therapy, you can go to second-line treatment. This is a second therapy that is also demonstrated to work. But at one point, you might exhaust all the available therapy. So at that point, your treating physician will recommend you to a clinical trial. Most of the time, if they don't have clinical trial in their organization, they will refer you to a university affiliated hospital, which most of the time, that's where the clinical trials are. Patients should do their own work also. I always say that we can work at all different places, but we are the CEO of our body. So we need to decide what we want to do with our body. So there is a lot of tools that are available for patients. Some of them you can go on clinicaltrial.gov and you'll have all the clinical trials that are going on in the US. But what I highly recommend also is to go to patient advocacy group. So if you have lung cancer, you can just type, lung cancer patient advocacy group. And those are people that have the same journey than you. Those are people that had cancer and they did their own work and they have supporting group. So I think that I always recommend people to go to those groups and they are aware of the next treatment. They are coming to pharma and they are asking us what is next. So those patients are very well educated and they're a great resource for people who have cancer. And it's a shocking experience. It's something that it's very difficult to go alone. So if you want to go to those, it's up to you. You don't have to do that. But if you're asking me what to do, I think it's a wonderful way to go through that journey.
That is very useful. Thanks, Sylvie. So we have another question about how far away is AI from being used in mainstream and early diagnosis? It's
there. I think that it might not be a way that it's approved this way, but I think we're using it. I think that we're at this point, sometimes we're looking at different mutation, and we can see that patients that have this mutation and this concomitant condition, maybe it's better to use that treatment. So I think all of that it's on its way, and it's going to be used more and more in the future. I think that it's going to be, all those programs are going to be made user friendly, and physicians are going to use them constantly. and exchange information also to make those tools even smarter as we get more data to improve them.
We have another question. What are your thoughts on using AI/ML to be more inclusive in terms of serving patients around the world? And I think that with clinical trials, we're trying, I think it's an aspiration to be more and more inclusive.
Absolutely. And I think that we are really trying very hard in clinical trial to be more inclusive, include more minority in our clinical trial. And in fact, unfortunately, a lot of the data that we have, or the genome sequencing and the information, is also very a selected type of population. So we're trying to collect that information so we can have more information on different ethnicity, different group population, to really be able to serve everybody that we want to deliver the drug. So AI, the same way, we have to collect that information for all different patients so we can have the information to also to serve them appropriately.
Thanks, Sylvie. I think that we've answered all the questions that people have had. So I hope that this has been useful to everyone. It certainly has to me. I think it's important for everyone to know where we are in technology and in science and how hard we're working in order to really make people's lives better and give them a greater chance of a good prognosis. And I think it's an important message to, if you have been diagnosed with cancer, don't stop fighting. And advocate for yourself and try to learn as much as you can. Join those patient advocacy groups and try to find out if there is information on clinical trials that your physician might not know about, perhaps. And there's new trials, I think, coming out at a greater pace now because of AI/ML. This has really sounds like it's been incredibly impactful and changing to the industry. And I'm very happy to know that. So if there are no more questions, I thank you so much, Sylvie, for sharing your experience and your expertise with all of us. And thank you everyone for joining.
Thank you so much. It was my pleasure. And I want to thank everybody to do their contribution in really helping us to do and develop better medicine for the patient. So thank you so much.